I'm Kathrin, CEO of OptoBiolabs, and it's my pleasure today to present to you our most recent findings of the ATTRACT Phase 1 project OptoCS, Optogenetic Cell Sorting. Our consortium consists of the University of Freiburg, the EMBL in Heidelberg, and the startup company OptoBiolabs. Together, we are developing an illumination device for optogenetic cell sorting. Let me briefly introduce you to optogenetics and why it is relevant. You genetically engineer any given cell sample you want, and you render it to become responsive to light. This way you can shine, for example, green light on a given cell sample to induce cell division, or shine red light on the exact same cell sample to induce cell death. Think about what this could do to personalized medicine and other applications. In optogenetic cell sorting, cells are being continuously taken up from our temperature-controlled tube illuminator. The cells then travel along a capillary towards the cell sorter. The OptoCS prototype illuminates these cells along the way to the cell sorter to induce an optogenetic effect. Here we see an increase of GFP. The responding cells are then selected by the cell sorter and can be used for various downstream applications, like single-cell sequencing, cell line development, or personalized CAR T-cell therapy. Since October, we managed to completely redesign our sample tube holder and develop a special illumination platform that enables super flexible illumination along the capillary. What we have left to do is putting all the single pieces together to one fully functional OptoCS product. Since 2018, we have been pretty busy gathering funds like the Exist and Young Innovator Startup fundings. And we launched our first product, the PX1 for optogenetic flow cytometry. We won business plan competitions and were listed MIT Innovator Under 35 Europe for our first product. The ATTRACT funding and collaboration with the EMBL and University of Freiburg enabled us to build the first OptoCS prototype and test it at the EMBL in Heidelberg. The idea of optogenetic cell sorting attracted our first investor, Matthias Bissinger. We are already talking to the leading cytometry companies to collaborate and drive optogenetic cell sorting further. Together with the ATTRACT Phase 2 funding and network, we are certain to be able to launch our OptoCS product in 2022. I would like to acknowledge the people who actually did the work. Not just the team of OptoBiolabs, but especially Malte and Diana from the EMBL. Without their technical expertise, this project would simply have been impossible. And of course Sasha from the University of Freiburg, who offered his expertise in optogenetics and access to his cell systems. With this, I would like to thank you for your attention and for giving us the chance to develop high-risk technical devices for optogenetic imaging. If you have any questions, please visit our homepage or write to info@optobiolabs.com. Thank you very much.
Hello, and thank you for the opportunity to present the recent developments in the 3D Meta ATTRACT project, where we aim to develop dynamic metasurfaces for 3D imaging. We hope to achieve fast and wide-angle beam steering at high resolution through the combination of MEMS and metasurfaces. This platform poses several advantages over current state-of-the-art LiDAR technologies based on MEMS, micromirrors, gratings, and phased arrays. Here we can see a picture of the device that we fabricated last year. On a PCB, we have placed a piezo MEMS micromirror developed at SINTEF in Oslo. And on top of this, we have a glass substrate where we have put a metasurface that has been fabricated by our project partners at the University of Southern Denmark. The way that this works is that by applying a voltage to the micromirror is brought close to the metasurface. And when we do that, we change the optical properties of the metasurface. For the application of LiDAR, we are interested in this one, where light is sent into the metasurface and then sent to another direction. And when the metasurface is pulled away, the light is just sent back to the same direction. But we can also use it to, for example, make a lens which you can switch on and off. Here we see the measurement. At 0 voltages, the light is sent back in the same direction it came, in the 0th diffraction order. But upon applying a voltage, the light is sent to the 1st diffraction order. So today I'm going to talk about what we've done since October last year, and in summary, this is what we've done. We've demonstrated novel functionalities with the platform and have made additional measurements. Using these measurements, we've sent a manuscript for publication in Science Advances. We've also secured funds for a collaborative PhD between the project partners, SINTEF and SDU. And currently we're working on identifying other ATTRACT initiatives within LiDAR, whom we hope to collaborate with for phase 2 of ATTRACT. So here we can see the metasurface work in practice, switching at a speed of 1 Hertz between the 0th and the 1st diffraction order. Since October, we've characterized this better and measured the rise and fall times to be 0.3 and 0.4 milliseconds. This is, of course, based on the current system where the micromirror is much too big compared to the MEMS, and this can be dramatically improved by making the MEMS smaller. We expect maybe on the order of 100 to order of 1,000 times improvement. And this is really one of the strengths of the platform, that we can get very fast switching, which by using this within a LiDAR system can give a very fast continuous scanning or a very fast frame rate, while at the same time having high resolution. For the article, we decided that we wanted to demonstrate the versatility of the platform, and so we did that by also implementing a lens function. As we can see here, at lower voltage, nothing happens to the light, but upon increasing the voltage, the beam is focused to a point, acting as a lens. We've also worked on scaling up the metasurface to give us several diffraction orders instead of only 2. And we have some interesting ideas on how to do that for phase 2. As mentioned, we've submitted an article to Science Advances. It's currently in review, and we're quite hopeful that it will be published. We're very happy that thanks to ATTRACT, we were able to demonstrate the principle, and we were able to secure internal funds for a PhD collaboration with the nano-optics group at the Southern University of Denmark. And our colleague Paul has since the 1st of January been working as a PhD student to develop the platform further. I just wanted to say one thing about the next steps. This is something that we covered in the report last year. But of course, in order to develop a LiDAR for autonomous driving, there are many components that need to be improved. And we've identified many ATTRACT initiatives within LiDAR. So we hope to contact them and hopefully find some good collaborations for phase 2. So thank you very much for your attention. That's about what I can reach in 5 minutes. But if there are any questions, don't hesitate to contact me. I'll be happy to answer. Thank you very much. Bye-bye.
"3D Cancer Spec". This project is about technology transfer from astronomy to medicine and potentially a disruptive innovation for cancer diagnostics. The main point is that astronomy has developed a technique called integral field spectroscopy, which is like taking an image with a camera, however a full spectrum in each pixel rather than only an intensity level in each pixel. In combination with illumination of tissue with a laser, this technique can record a full Raman spectrum in only one exposure, point by point. Raman spectroscopy has been shown already 10 years ago or so to be able to distinguish malign from benign tissue. However, sequential point-by-point scanning turned out to be impractical for medical doctors and their patients. The major breakthrough of taking Raman images in one shot only will enable surgeons to determine lesion margins directly through computer-enhanced visual inspection during an operation, so enabling the removal of malign tissue in only one step, without the classical preceding biopsy. This would mean an enormous ease for the patient and potentially a higher success rate for the surgery, securing the entire removal of cancerous tissue. Our clinical partners have convinced us that this would mean a truly disruptive innovation. As for the current status of the project, we have conducted a complete end-to-end design study, including costing, such that we could, in essence, start immediately with the manufacture of a prototype, which would indeed be the goal for ATTRACT Phase 2. All the essential elements are at hand: a core group to form the consortium, the optomechanical design of the spectrograph, the optomechanical design of the fiber sensor, the detector system, and also the necessary software, data reduction and analysis. Concerning the next steps in ATTRACT Phase 2. First of all, building the prototype will be a prerequisite to conduct clinical studies that are necessary to fully validate the technology and provide certification in agreement with European law. Secondly, the core group, consisting of the company Winlight System in Pertuis, France, and the Leibniz Institute for Astrophysics, AIP, in Potsdam, Germany, would need to be enhanced by an academic partner for the interpretation of multimodal data, with an industrial partner for the fiber sensor, and also with hospitals for the envisaged clinical studies. As we have already published in the literature, the immediate first application would be skin cancer diagnostics. However, we have already begun experiments with endoscopy towards application for cervix and bladder cancer, and there is also a very interesting application for the detection of plastic microparticles in human tissue. More applications are certainly to come. For the further development of the consortium, we will make full use of our networks, for example, the European Photonic Industry Consortium EPIC, OptecBB, 3D Sensation, Leibniz Health, and the corporate connection to the Sinem Group in France. Furthermore, we are in the process of broadening the scientific scope of this unique application. This means that we are continuing to apply for grants covering certain aspects of the research that is necessary to further develop the methodology beyond the mere technical development of the system. However, we have already been contacted by a venture capital company in the Netherlands who does see the potential that we are developing, so we are convinced that the prototype will materialize. Let me say that it would be a shame if we would miss the opportunity of this European lead in imaging spectroscopy and let other parties commercialize this exciting technique. Thank you for your attention.
Hello and welcome to the update for UTXmCT, unstained targeting, X-ray phase contrast micro-CT as a bridging imaging modality for intravitreal CLEM. Following up from October, we wish to report we completed point 3, validating the imaging system on an extremely challenging biological specimen. This is a Drosophila larva, widely used model in biology and the life sciences. The sample was prepared without any staining, and you can see in panel A the image acquired at a synchrotron facility, and in panel B the image of the same part of the sample acquired with a custom system we built for this ATTRACT project. A is a tens of millions of pound instrument, an undulator in a third-generation light source and a 200 meters long beamline. B is an X-ray tube-powered tabletop system in the range of hundreds of thousands of pounds. This is a 2D slice extracted from a complete 3D volume image. A is our gold standard, and we are happy to report that we were successful in producing a volume image that enables navigating the anatomy of an unstained soft tissue sample with our proof of principle system. It is possible to recognize major anatomical landmarks, as indicated by the red arrows. Those can be located with micron-scale precision in a volume of approximately 1 cubic millimeter. These are the elements required for targeting specific groups of cells or tissue structures in multicellular organisms for subsequent analysis and imaging at the nano and atomic scales. Just to be clear, we don't outperform the synchrotron. We cannot possibly do that with an X-ray tube. But the point here is that, of everything that is there to see in this sample, panel A, our system is capable of providing a representation of the sample with enough contrast and details to enable targeting without the need for staining. And here's an update on partners. Since October, we continue discussions and there are 2 more corporations and 1 more academic institution joining our project, further expanding our consortium for X-ray generation and beam conditioning, fabrication and design respectively. Any question, please do not hesitate to get in touch at m.endrizzi@ucl.ac.uk. And thank you very much for your kind attention.
Hello, I'm Rok Dolenec, speaking on behalf of the Real-Time Fluorescence Lifetime Acquisition System, or RfLAS for short, project from ATTRACT Phase 1. I'm glad to be able to report to you the progress of this project since ATTRACT Phase 1 wrap up. It has generated a lot of excitement in the group of researchers working on it, and also between the potential users of this technology that we have established contacts with. Since October last year, we have actually never paused working on the project, but let me summarize all this progress just in the next few points. We have been working on the hardware of the prototype system, modifying it to enable tests for various applications, explore how we can improve it, and test what is the actual performance of the system. Most importantly here, we have tested different target molecules or fluorophores, and also at lower concentrations. This is important because it showed us how the system would perform in a much wider range of possible applications, and I'm happy to report that the system performed just as well as before in all these various scenarios that we have so far tested for. More interesting may be that we have started an ecosystem of potential users. So these were people that we found in various institutions, universities, companies, that have expressed an interest in testing our system or contribute to its design. And these various communications range all the way up to actual official letters of support that some of the most interested parties already prepared for us. And one of these companies is a pharmaceutical company that could potentially be our first big customer, of course, once the technology reaches a sufficient level. In this time, we have also obtained additional financing that will enable us to produce 3 small but independent and portable prototypes that we can give to potential users that are interested in using it, and they can then test it in their environments and their applications. And this will give us a very valuable feedback, especially on how we can optimize the system for their specific applications. For some time now, we are also working on securing the intellectual property. I can say that we are in process of patent application, but this is still ongoing. So we're at a stage that we are looking for the next round of financing that would enable us to move the system to the next level. There we could create systems that are optimized for specific applications of the users and actually move towards production at scale. And we are very full of ideas what we can do with this technology, where we can move forward. And we're looking forward to see how we can continue to contribute to the ATTRACT story all the way from the very beneficial phase 1 now finished, but also in the phase 2. And we believe phase 2 could be a perfect opportunity for this project to grow further. Thank you.
Good day to everybody. In this short presentation, I will show you the results we obtained with our ATTRACT project, which is called Graphene Gallium Microcell Arrays for Color-Sensitive Terahertz Imaging Sensors. The acronym is GRANT. The idea of this project was to develop a color-sensitive terahertz imaging detector based on graphene technology. Graphene-based Gallium microcell arrays will be realized by a combination of micromachining, plasmonic, and graphene technology. This device, if successful, will be a core or a revolutionary light, portable, and autonomous terahertz inspection system. I will just go very briefly through all the results we obtained during the project. So first of all, the first problem we faced was to realize graphene membrane, which we were able to cover not only with single Gallium cells, but an array of Gallium cells. And we were able to obtain this task by combining titanium with graphene. So adding a thin layer of titanium to a graphene layer, we were able to obtain large areas like this one, which is 12 by 12 pixels. Each one is 20 microns, so that means it's something like a 300 by 300 micron array of detector. Second point, the second task was to obtain a good sealing of these Golay cells in order to maintain the pressure inside even when the pressure is increased. And here you can see that we were able to, our membranes were able to sustain a pressure difference of about 1 atmosphere. And we were also able to cycle continuously pumping and down pumping of the membrane without observing any leak of the gas side. Moreover, the addition of titanium resulted to be very, very useful to create a more sensitive membranes. Actually, what happens when you add a few nanometers of titanium of a graphene layer, this apply a stress on the graphene layer, which is released when the membrane is suspended. So it creates some wrinkles that can buckle during the transfer, but only in suspended areas. That results in a membrane that can be easily inflated and so have a higher transduction of the difference in pressure, so in the temperature, which is the base of the process in the Golay cells, into something which is easily detectable. This is, here you can see a single graphene without titanium, which is very flat after transfer, and this is graphene with titanium, which is buckled and wrinkled after transfer. This change doesn't affect the mechanical properties. Here you can see the indentation and you can see the breakout of the membrane when the pressure, the local pressure, exceeds 400,000 Pascal. Once we had the membrane ready, we were able to produce the Golay cells and to test the Golay cells. These are the Golay cells which are tested with this line, with a 5 terahertz line obtained out of a global source, which is a broadband infrared source, but we were selecting only the 5 terahertz, and we observe the response. So the Golay cells are able to sense terahertz radiation. This is a very low power, it's a few microwatt per centimeter square, but unfortunately it's slow. So we have response in seconds and it's continuously increasing. So we have to engineer this effect with a differential measurement. And the differential measurement can be obtained not as the demonstration experiments that we've seen with dark field imaging, but with a different alternative scheme of detection which we developed, which is based on self-mixing. So the laser, the light emitted by a laser is self-mixed with a reflected light from the membrane, and that can give us a resolution which is of the order of few nanometers. Here you can see the interference through position change in the membrane, graphene membrane, of a few fraction of micron. Here you can see a map of the membrane obtained by self-mixing scheme. While we're using the self-mixing scheme, we also apply this to another way to detect the terahertz radiation, which is based on oscillating membranes, silicon membranes that change their resonance frequency while oscillating, so they can be used to absorb resonance frequency and get the results of the frequency. This showed and demonstrated a very high time resolution, which is about 20 Hertz of cutting frequency, but still need to be improved because for the moment it works in vacuum condition. With this I conclude. Let me just thank you for your attention and all the people that collaborated to this project.